What's up, science fans? Now, I know many of you enjoy learning about human biology and development, how we all start out from a single fertilized egg and slowly become complex beings. Well, today we're tackling a stage of development that has long mystified researchers, the pivotal process known as gastrulation. So, what exactly is gastrulation? Well, in simple terms, it's when the blob of replicated cells that make up an early embryo starts reorganizing itself into distinct layers, setting the stage for our cells to become specialized and form organs and tissues, kind of like renovating a house by adding new walls and rooms. This process happens in humans around 2 weeks after fertilization. But here's the thing. Scientific guidelines say we can't study natural human embryos in the lab past 14 days for ethical reasons. So gastrulation has been like a black box. We know it's important, but haven't been able to peek inside. Trust me, developmental biologists have been itching to unlock the secrets of gastrulation for decades now. How exactly do our cells know how to rearrange themselves? What genes and signals are involved? Are there any similarities to gastrulation in other species like mice or frogs? All very probing questions. Well, thanks to some ingenious work by researchers at UT Southwestern, it seems we finally have a way to study human gastrulation in action for the first time. In embryo-like structures grown in a dish. Let's dive deeper into the science behind this breakthrough. All right, so let's dig into the nuts and bolts of how the researchers pulled this off. Their secret weapon was using these special cells called expanded pluripotent stem cells, or EPSCs for short. Now, you've probably heard of stem cells before. They're like blank slates that can become any type of cell. But EPSCs have some unique superpowers that other stem cells don't. One is that they can generate both embryonic cells, the kind that will become the fetus, as well as extra-embryonic cells, which form vital supporting structures like the placenta and yolk sac. This was crucial because previous embryonic models totally lacked those extra-embryonic tissues, and they provide essential nourishment and signaling factors that guide development. So without them, prior models were missing a big piece of the developmental puzzle. By exposing the EPSCs to just the right cocktail of growth factors, the team was able to coax them into forming tiny structures nearly identical to a 3-week-old human embryo, along with an attached yolk sac. They call these structures perigastruloids. Because perigastruloids contain both embryonic and extraembryonic cell types, they could truly mimic the complex morphological changes and tissue interactions that happen during gastrulation and early development, something previous models just couldn't achieve. Now, the researchers didn't just create 1 or 2 perigastruloids, they pumped out batches of hundreds. This suggests the EPSCs have like a superpower to consistently self-assemble into proper embryonic structures with minimal input. The scientists did have to give them a nudge with the right growth factors, but besides that, the EPSCs took care of the rest. No complicated equipment or micromanaging required. Of course, the research team made sure to follow ethical guidelines and stopped the perigastruloid growth after 3 weeks. We don't want any mad scientist situations here. Even within that limited developmental time frame, though, the perigastruloids could mimic intricate morphological changes that take place during gastrulation and early organ formation better than any previous human model. Now, the perigastruloids did have one weakness. They weren't viable long-term because they lacked certain placental components. Think of them like Tamagotchis. Remember those? They needed regular care and feeding by researchers in the lab to survive. But the study authors are already working on ways to improve viability by incorporating more cell types. So in the future, we may have even better perigastruloids that can develop for longer periods and reveal even more secrets of our embryonic beginnings. Now, so far, we've focused on human EPSCs, but scientists think this technique could potentially also work for EPSCs from other mammals like mice or even monkeys. Of course, more research is needed to confirm if perigastruloids can be generated from other species too. As for functionality, the human perigastruloids did sometimes display spontaneous twitching movements, almost like very early primal reflexes. Creepy, but also really cool from a developmental biology perspective. The researchers did observe a small percentage of abnormal perigastruloids with structural defects. These mutants could actually provide valuable insights into what can go wrong during early embryonic development and lead to birth defects. In the lab, the scientists are using perigastruloids as a new model system to study milestones like gastrulation, body axis formation, neurulation, and early organ growth under a microscope day to day as they happen. Looking forward, there's huge potential for perigastruloids to be used for screening drug or toxin safety to see if they negatively impact early development in a dish before clinical trials. All right, we've covered a ton of ground here. Let's bring it on home and recap this amazing research, opening up the mysteries of gastrulation. Thanks to their brilliant use of souped-up EPSCs, the scientists created living perigastruloid models that finally reveal how our embryonic cells cooperate to transition from a blob into the first rudimentary bodily structures and tissues. Huge win! It's like we finally got our golden ticket into Willy Wonka's super-secret lab where human gastrulation happens. Mind-blowing stuff for development nerds like me. Of course, this is just the prequel. Adding more cell types could make perigastruloids viable for longer and enhance our biological blockbuster sequel. Just imagine screening future drugs right in the dish to see if they make our perigastruloid cells go haywire during gastrulation. Talk about a nail-biter plot twist. The point is, this changes everything. We've breached the black box of development, and I'm pumped to see what awesomeness these models unlock next about our origins as embryos. Catch ya later! If this seemed interesting to you, this is only the beginning. There are so many interesting topics to cover, and so much to learn.